After this little exercise, I'd like to collect some feedback from you. Then I'm going to ask you if you've got questions about the Socrative quizzes that we took last class, or the exercise. We'll wrap up the exercise that you spent all class last time on Friday doing. So it's the codon table. What do we do with the codon table? What's the purpose of the codon table? What do we do with the codon table? Spell have a nice day.
To get amino acids.
To get amino acids from an RNA sequence. So you take codons, right? So there's a codon, AGA. That's in RNA. So you've got an RNA transcript, it's got a codon. What amino acid does it encode? First letter, second letter,
Wait, that's our what?
third letter.
Wait, you said that was an RNA? sequence?
Yeah, that's RNA. You can't tell, I'm just telling you it's RNA.
Okay.
How would you know it was RNA? Unless if I don't tell you, what would clue you in?
Yeah.
Yeah, uracil. So if you see something and there's U's instead of T's, then it's definitely RNA. But there's no T's or U's in that sequence. That's why I had to tell you this was RNA. Plus it's single-stranded. But sometimes I write DNA as a single strand and not as double-stranded, so that's not really helpful either. So AGA, if that's in an RNA molecule and a ribosome is sitting on top of that, small subunit, large subunit, and it's going to translate that codon into an amino acid. What amino acid is it? Yep, AGA, arginine. So that transfer RNA molecule that's attached to that clover leaf would be recruited by the ribosome and stuck onto the C-terminal end of a growing protein. What's weird about the codon table? What's unique about it? What's expected, what's unexpected? What patterns do you see? What features do you see in?
If you look at A, C, it doesn't matter what the third.
If I look at AC, it doesn't matter what the, okay, AC, doesn't matter what the third nucleotide is. They all encode the same amino acid, threonine. Is that true for all of the codons? Okay, so what's the opposite of that, essentially? Like, find one that doesn't do that. Which one? CAU. So we've got histidine. It's only encoded by two different codons, CAU and CAC. AUG, methionine, our start codon, that's the only codon that encodes methionine. So you've got some amino acids where you can have a number of different codons, each encode the same amino acid, and you've got some codons where there's just one. It's unique, that's it. If you don't have those three specific nucleotides in a row, you don't put that amino acid in the protein. What's the largest number of codons that can code an amino acid? Is it 4? So we're like alanine's got 4, threonine's got 4, proline's got 4, serines got. So leucine's got 6. Serines got 6. Arginine's got 6. So there's a variety. That's the point. You don't need to memorize which ones have 6, which ones have 4. But the point is that there's variety. Some amino acids are only encoded by a single codon. Some are encoded by up to 6. Does that tell us anything interesting? Why would there be those sorts of differences?
That would happen, like, for example, if there are mutations, like single nucleotides, some of them wouldn't matter. For example, if we're looking at a mutation in our gene, and it would be CGA, and we changed the third A for a G, it wouldn't actually affect the protein. Right.
So if you mutated CGA to CGG there, if you had that specific point mutation, it wouldn't change the amino acid. So sometimes this redundancy is useful. The redundancy is required. I mean, it has to be. How many different essential amino acids are there?
20.
Right. So if there are 20 amino acids, and we use codons, how many codons are there? 64. Yeah, there's going to be on average 3 codons per amino acid. There's going to be some redundancy. That's just the math. We've got way too many codons for the number of amino acids that need to be encoded. So that's some basic features of the codon table. Sometimes you can have mutations that don't change the amino acids. Sometimes mutations in the gene would change amino acids. Does anybody have any questions about the Socrative questions on translation that we had for Wednesday? You might not even remember what they were.
No, we do not.
All right. A lot has happened since then. So there was one. Types of RNA involved in translation. What gets translated? Messenger RNA. What does the translation process? What's the protein that does translation? It's the ribosome, so we have to have ribosomal RNA. That's part of the ribosome. And what brings the amino acids to the ribosome? tRNA. Okay. So it's those 3. Messenger RNAs, ribosomal RNAs, and transfer RNAs all take part in translation.
Are the other ones like a thing?
The other were things, but they're not things related to translation. The distractors are real types of RNA, but they weren't relevant. Okay, this one we have to go over because there was still a variety of answers for this question. Ribosomes add new amino acids, like I just drew. Transfer RNA with an amino acid on it comes into the ribosome and gets stuck onto the C-terminal end. No 5', no 3'. That's nucleic acid terminology. That's not proteins. So it's just C in this case, just C-terminus. Let's see which question this was. Oh, you basically got that one. Methionine is the first amino acid of every protein. Don't have to add that picture in. Let's see, what's this one? Ah. Yeah, I know, true-false questions are horrible. This one got mixed results on the quiz anyway. So does the small subunit of the ribosome in eukaryotes bind the 5' cap, scan along the RNA molecule reading until it reaches the first AUG? And then it starts translation. Is that how it works? What? No, I'm not talking about capping. So transcription, what happens, what's the first step in transcription? So RNA polymerase starts on the template strand of DNA. And what's coming out? RNA polymerase. Yep, and then that 7-methylguanine cap gets added. And then after transcription is complete, that molecule of RNA gets sent out of the nucleus into the cytoplasm, where it gets translated. So the question is, so there's a 7-methylguanine cap on a spliced and also poly-A-tailed mature messenger RNA. And that's what gets us to this setup for this question. So once the processed messenger RNA is in the nucleus, and it's going to be translated by a ribosome, is that statement true? Yes. That's exactly what happens. And this, you please, please, please have to remember. On Friday, if everything else leaves your head during the exam, this is the one thing that you should remember, that in eukaryotes, how does the ribosome know where to start the process of translation of a transcript? The start codon. It's the first start codon that you see in a transcript. So imagine this is the sequence of a transcript. Don't imagine. This is the start sequence of a transcript. I'm just making stuff up. Okay. What's the first amino acid that's encoded by this RNA molecule? Somebody in the back half of the class has to answer. Don't even look at what I just wrote. What's the first amino acid that's encoded by a transcript? What's always the start codon? AUG. What amino acid does it encode? Methionine. Okay, so the first amino, regardless of what that says, don't worry about that. Okay, where's the AUG? It's near the end. There's AUG. So what happens is, and the reason this answer is true, is that here's a 7-methylguanine cap. That ribosome small subunit binds there, and it reads this direction until it spots that first AUG, and that's where it starts saying, okay, there's an AUG, there's my methionine. That's where it starts reading codons. And then it reads in groups of 3, moving that direction down the transcript. So please, 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 there will be questions where I ask you to do this and translate messenger RNA sequences using the codon table. Do not necessarily start translating from the first 3 letters you see. In this case, GUA. Because that's not a start codon. If it's eukaryotic, you have to look for the first AUG, then start translating. Okay. If you don't take anything else away from today's class, please write that down 3 times and remember it. It will save you on the exam. Now, it's different from how prokaryotes do translation. We've talked about that in class. You can go back and review the notes to figure out how they do it differently. But eukaryotes, small subunit binds the 7-methylguanine cap and reads from left to right, 5' to 3' until it reaches AUG. Then translation starts. I think there was one more question. Any other questions while I'm loading up this last question from the quiz? That's a good one. Just so you know, and just so it's recorded in the notes, asterisk is how stop codons are represented. So if you have AUG, GGG, UGA, anybody got a codon table out? What's AUG? Methionine. What's GGG? It's G. That's the only one I can remember. Glycine. And then UGA is a stop. So it's represented in single letter code by an asterisk.
There's 3 of them, right?
There are 3 stop codons, UGA, UAG, and UAA. So any of those 3 will cease translation. If a ribosome reads one of those 3, those are the stop codons. Yeah?
So, how methionine is the start codon, but
Yes.
it also represents an amino acid. Does the end codon represent a certain amino acid, or is it just N?
So the stop codons, the 3 stop codons that end translation, there is no transfer RNA that matches those. In other words, there is no amino acid that gets inserted when there's a stop codon. So in this case, the N-terminal amino acid is what? The amino terminal or N-terminal. What was the first amino acid that got incorporated? Methionine. So that's the N-terminus of this protein. There's a methionine, it's hooked up to a glycine. What's the C-terminal amino acid? Glycine. So this is a 2 amino acid protein. Then nothing where the stop codon is. Great question. So 3 codons, 2 amino acids. 50 codons, 49 amino acids. Stop codon doesn't have an amino acid.
When you add the cap and poly-A tails, we have to know the enzymes?
You don't have to know the enzymes that add the cap or the poly-A tail.
So you can just say they get added?
Yes, you can just say they get added. Yeah.
What happens to the mRNA after it's been used?
Ooh. What happens to the messenger RNA after it's been translated? Once?
Once, or is it used more than once?
It's used more than once. That's the fantastic thing about transcription and translation. One of the things that I didn't tell you yet, but now you get to know. The reason that it's beneficial to do transcription and then translation is not only to get the message out of the nucleus into the cytoplasm, because translation in our cells doesn't happen in the nucleus, so we have to get nucleic acid out of our nucleus into the cytoplasm for it to be translated and turned into a protein. The other benefit, though, is that when you transcribe a gene, how many times can a gene be transcribed, read and a messenger RNA made? Does our chromosome get used up when it gets transcribed? No. So you can make lots of RNA molecules from a single gene. And then all of those transcripts from the same gene go out into the cytoplasm, and each of those can get translated multiple times by ribosomes, read over and over and over and over again by ribosomes, and you make lots and lots and lots and lots of protein. In some, it's signal amplification. You start with 2 genes, you're diploid, you've got 2 copies of the gene in your nucleus. You make loads of messenger RNA from those, send them out into the cytoplasm, and then you've got lots of ribosomes in the cytoplasm that read all of those RNAs and make lots and lots and lots of proteins from every messenger RNA molecule. And then at some point, the messenger RNA molecules get degraded. So they do have a finite lifetime. And that's something that you'll probably talk more about in cell biology if you take 103, is the mechanisms inside our cells that control how quickly messenger RNAs stick around. So their lifetimes are not forever. They do get degraded eventually. Other questions?
Can you maybe review green point mapping?
No, that I'll do on Wednesday. Yes, so bring that question on Wednesday. Try to do translation. Thank you, though. Yeah. Wednesday review session. Today, we try to finish translation. So this was, as I understand, a summary of your findings from our little exercise on last Wednesday, taking the NUO1 DNA sequence, making random mutations to it. So you were my mutagens, randomly screwed up NUO1. And I was actually kind of surprised that you didn't get X's down here in all of these boxes. So the take-home message, which I'm sure you talked about on Wednesday, should have been any sort of mutation can make any of these types of effects on a protein. So you can't just take an insertion mutation and know that it's going to have a particular effect on the protein. You actually have to do the process of translation to figure out what the effect of the mutation was. Is there one of these combinations that actually is impossible? So an insertion of 1 nucleotide, an insertion of 2, an insertion of 3. There's a lot of gaps here. Is one of these things that you didn't observe on Wednesday actually not possible to do? Is it possible to insert 3 nucleotides into an exon and not change the protein sequence? So here's an exon. 3 codons. 3 amino acids. What's CCC? Proline, I think. Sorry, that's not 3 amino acids. 3 codons, 2 amino acids. I think that's proline. And stop. Okay, so can we add, yeah, question?
No, I was gonna, sort of a question, but, if you were to like add 3, acids that made up an N codon right at the end before the N codon, would that be exactly the same?
Ah, yes. So the question is, can you insert 3 nucleotides down here? We're just going to pick on this one. It's a thought question for you. I'm not going to do this for the whole table. Is it possible to insert 3 codons and not change the protein sequence? So that's an excellent advice, or an excellent suggestion. What if we stuck 3 nucleotides in right there, between the second and the third codons? What could we add that wouldn't change the protein length? Yeah, another UAG, a UGA, one of the stop codons. UAG. So now we've inserted 3 nucleotides into an exon, didn't change the number of amino acids it encoded. Same protein, didn't affect the protein at all. Okay, great. Very good. So you might spend some time thinking about which of these other gaps in this table, which of them literally cannot be done, which of them we just didn't come up with examples for in class. Okay. Time to practice what I preached. Here's the practice question for the exam. Okay. 2 transcripts. Messenger RNA molecules, I'm not showing you the 7-methylguanine cap, but here they are. Underline the codons. The sets of 3 nucleotides that the ribosome is going to read. Talk to a neighbor and make sure you agree. What about a minute to do this? Okay, that's it. Enough time for underlining. What questions are you asking each other right now, if you haven't figured it out already?
Do we skip the first letter?
Do you?
Or the last, or do we ignore the last one?
So for that first line, what's the first codon? AUG. For the second sequence, what's the first codon? AUG. So then you go from there in groups of 3, and the question that was just asked was, what happens if you run out of letters? Then there isn't a codon. But in this case, I've got groups of 3. There's 2. Does it matter to us that there's a second AUG there?
No, you said
It's a start codon, but it's also just the codon that encodes methionine. So it's okay if there's an AUG in your protein, that's fine. A lot of proteins have a lot of methionines. So that doesn't mean it's a start codon. It is a start codon in that it encodes methionine, but it's not the first one. So it's not the start codon, it's just a methionine codon. Are there other concerns, issues, questions you have about these? Yeah?
there's 2 stop codons with first transfer.
Okay. Where are the stop codons?
UAG.
So there's a stop codon.
There's 1 to the left.
Where, this guy?
Yeah.
Okay, this is the perfect question to ask. Thank you. By the way, what about the second line? Underline all the stop codons in the second molecule, and then we'll answer that question.
It's the last one. The very first one.
Is that it? Yeah. So, does this cause anybody any consternation? So what's the deal with this UAG? That's a stop codon, except it's not in the correct reading frame. That is, it's not a triplet that the ribosome is going to read, because we underlined those in blue. Those are the triplets, the codons, that are going to encode the amino acids or the stop codons. So this UAG is what we call out of frame. It's out of the reading frame, which means that even though the ribosome is going to read past it, it's not going to stop on that UAG and say, oh, it's a stop codon, because that ribosome is reading in successive groups of 3. Likewise, what about that bottom sequence? The first 3 nucleotides are a stop codon.
Doesn't matter. It'll keep looking for the UAG.
Doesn't matter, because the ribosome binds the cap and scans until it hits the first start codon. So the ribosome binds there and scans right past it until it hits the first AUG. Only then does it start caring, not that it cares because it doesn't have a brain, about which stop codon is in its reading frame. So we'll have a methionine and then some amino acid, amino acid, amino acid, amino acid, amino acid, and then stop. The first stop codon that was in its reading frame is what terminates translation. Yeah.
Would you want us to keep underlining the triplets after the stop codon?
You don't have to, no. Anything after the stop codon, you don't have to worry about. Yeah.
Does the stop codon get translated, or is it stop translation before the stop codon?
So the stop codon, there is no amino acid that gets added. So the amino acid just to its left is the last one. So in this case, UGG, not the fabulous piece of shoe wear, but what's UGG? Tryptophan? UGG.
Yeah.
Sweet. W, tryptophan. UGG. Okay, there was a long list of stuff I asked you to do for class today at the end of class last time. It's all practicing translation. So I'm happy to try to do as much of this as you want, because all of these questions, I think, underscore important aspects of translation. But before we do that, if you've got other questions about translation up till now, it's a good time to ask. We'll keep moving forward if not. Got any burning concerns, questions? Okay. So by now you shouldn't need practice at translating this messenger RNA molecule. So this is the type of question I would ask. Here's a messenger RNA sequence. Sorry. I think that's my ringtone too. Find the first AUG, if this is eukaryotic, and then you underline successive groups of codons until we reach, somebody see if you can spot the first stop codon before I get there. Oh, there it is. Okay, so big messenger RNA molecule, those are the codons. Starts with methionine, ends with a stop codon, and then the point is you use the codon table to translate those codons. I'm not going to do that in class. So try this. If you don't know how to do this, ask during the review session or during office hours. Today, for an hour after class, and also remember Wednesday is the first day I'm switching my Wednesday hours to accommodate students who can't do noon to 1. I'll be doing just before class on Wednesdays, 10 to 10:45 or so in my office. And then I'll be walking over here. Wednesdays from now until the end of the semester. So bring questions on how to do that translation if you want. Oh, actually, sorry, I have to give you the answer. It's easy. For this one. This was the first mistake I made as a faculty member. I tried to create this really clever messenger RNA molecule that spelled map length, and I screwed up, and it spells map length. But then I realized, I turned it around, and I realized that this is a great opportunity to ask you to use your knowledge of how to use the codon table to design for me in this sequence a point mutation that will actually make that messenger RNA molecule spell what I was trying to, map length. So take a couple minutes to do that. Here's MAP-AUC. So that's an I now, and I want it to be an L. So 2 minutes. Look at the codon table, see if you can figure out how to change, make one nucleotide change that'll make my protein map length. Okay. Discuss with a neighbor first, and then we'll come back together in about 2 minutes. Make sure you got the right answer.
So we just need to change the name. He wants to create leucine instead of no, yeah. Well, he just wants to leucine, so he either wants to CU, CUA, no, or CUG, or but I guess there's the video.
All right. What are the possibilities for encoding L, leucine? What are the codons? So I'm going to circle them in the red there. It's just 6 of them. They all start with either U or C, but they all have a U in the middle, right? All 6 of those? Second letter in the codon is always U. So that's easy. That means there's no change from isoleucine to leucine. They both have U's in the middle. So what else? CUC. So we've made a single change. We changed the first letter from an A to a C. So there's AUC right here. We just changed that first letter, and we've made a leucine where there wasn't isoleucine. So that's what I should have done initially. I should have written CUC instead of AUC. This is common, by the way. The pattern that you'll notice, we haven't explicitly talked about, but now I'm going to. The pattern you'll notice in the codon table is almost always, if you change the first nucleotide in the codon, you change the amino acid. Remember we talked at the start of class about how the third letter, usually you can change it, and for most amino acids it doesn't matter if you mutate the third letter. Like here, proline, CC anything encodes proline. Mutate the third position, doesn't matter. Mutate the first position, usually changes the amino acid. So third position mutation's bad. No, good. First position mutation's bad. Is there any case up here where you can change the first letter and it stays the same?
Yeah, leucine.
Leucine, here, UUA and UUG are the same as CUA and CUG. So that's one exception. There's one more, arginine. Here, AGA and AGG or CGA and CGG are both arginine. There's 2 exceptions, but in general, don't mess with the first letter in the codon, if you can help it. Did that raise any new questions, concerns? Can I help? Okay. The rest of these questions that I've got in this set of slides, we're not going to get through. That's okay. These are all questions I asked in the translation video to watch for class last Wednesday. So I just wanted to provide the opportunity to address some of these questions. We're going to walk through them, but we don't have to get through them all. So one of the questions was, no, sorry, this was in the lecture slides, not the video thing. Is there anything missing from this messenger RNA molecule? So I'm claiming this is a messenger, this is a transcript. Messenger RNA, it's been spliced, and it's got a poly A tail. There is something missing. I haven't diagrammed on here. The cap would be one thing. It's got a start codon. Presumably it's got a stop codon. So that's, after adding the cap, that would be what an actual messenger RNA molecule would look like after it's capped, spliced, and tailed. It was missing a cap. Normally, and you might see this on the exam, I won't write 7mg. That's a good time. If you have a concern about that, raise your hand, ask me, I'll say it's fine. Okay. What I want you to take away from most of the rest of whatever we get through in the next 10 minutes or so is how changes in the nucleotide sequence affect the protein sequence. This is genetics, we're talking about mutations, but mutations to the DNA don't really matter unless the mutations, well, one of the main ways mutations affect us are mutations change proteins that are in our protein, amino acids that are in our proteins. Protein coding mutations that can really screw up phenotype, change phenotype. So what happens if we change the 6th nucleotide from a C to a G, the one that's in the box? Does this have an effect or not? Yes, because we, where was the start codon? Yeah, there was a start, the original start codon was there, but now this mutation has created a new start codon, AUG. So what's the effect on the protein, basically? Well,
It's shortened because the stop codon.
so if we, yeah, did it get started shorter, longer? Yeah, where's the stop codon? GAU, UAA. So now we have what used to be, I'm going to underline in black until I get bored. The original reading frame stopped there, UAG. That was the original stop codon. So that whole first line mostly was protein coding. The rest of it was 3' untranslated region. That's the part of the messenger RNA molecule that doesn't get translated because it's post-stop codon. It's after the stop codon. But now in blue, we've got just 3 amino acids. So this one change made a new start codon, shortened the protein sequence by introducing us to one of those originally out-of-frame stop codons, UAA. Right? Originally, that stop codon wasn't a stop codon because that's not a codon that the ribosome was looking at. But now it's a stop codon because it's in frame with the new start codon.
So does it start looking at frames of 3 once it hits the start codon?
Exactly right. So the start codon is what establishes the reading frame. So any AUG counts. And once the ribosome finds AUG, then it reads in the next 3, the next 3, and the next 3. So you have, honestly, you don't know what the reading frame is until you find that start codon. Great question. Okay, same sequence, different mutation. UAA to UAG. did we do? What did this mutation do that changes the protein?
Change the start codon.
We lost our original start codon. It was AUG, that was our start codon. Now it's GUG. So is there a start codon? Any AUGs in here? Or was that it? Second line, there. And then we've got codons starting from there. Oh dear. Look what happened. We skipped a stop codon, which is always before the poly A tail, so now we're just reading groups of A's. Translation has occurred into the 3' untranslated region, which is not supposed to be translated, why it's called untranslated region. So if the ribosome gets into the poly A tail, what's your protein sequence? What's AAA? Lysine. Lysine, lysine, lysine. Lysine's everywhere.
How does it disconnect them?
The ribosome just keeps going and going and going, and it falls off the end of the poly A tail, and yeah, I don't know.
So what phenotypic effect would that have?
Yeah, so how does this mess with your protein? Well, first of all, you get a protein that's totally different in sequence from the original. What was the original protein sequence? That was Mappy-Yanked, right? I think. I don't know. You should definitely practice translating this. I don't think it actually was. I think it was a different program I created for this. But this new protein starts with M. You do the translation on your own. So it's going to start with methionine. There's going to be some amino acids in the middle. Then there's a bunch of Ks. Totally different protein sequence than what the original protein encoded until this one point mutation. We've made a new protein, basically. And I can only guess at what the effect would be. First of all, you haven't made the protein that this gene was supposed to be making. It's probably bad. I can't say what the effect is because it's a made-up example, so I don't know what the original protein did because it was something I invented. The one thing I can tell you is that lysine is biochemists call it a sticky amino acid. I don't know exactly what they mean by that. They mean it gets tangled up with other proteins really easily. And that's a cause a lot of people think of neurodegenerative disorders like Alzheimer's disease, where you make long proteins that misfold and they tangle together and they make big masses of proteins in your neurons. And that's potentially one of the causes of neurodegeneration, the neurons start to die. So when you get strings of lysines, that might lead to that sort of an effect. That's just a guess, but hypothetical.
When the mRNA enters the ribosome, it goes from 5 to 3, and then it reads N to C.
The ribosome reads a messenger RNA molecule from its 5' end, the cap, to its 3' end, where the poly A tail is. And it synthesizes a protein that starts at the N terminus, the amino end, and goes to the C terminus. Please.
The mutations would happen after translation or after transcription?
All of these mutations I'm envisioning, although it doesn't really matter, are mutations in DNA that encode the genes that are then transcribed into this
But
RNA.
in that case, wouldn't the poly A tail and the 7G cap would be added in later? So we would actually see this happening. Because my question was, why would you have the 7 poly tail and the 7 the poly tail and the 7G cap if you haven't translated it yet?
So, translation. We've got the wild type, the original version, and you've got the mutant version of a DNA molecule. So, RNA polymerase is going to read both of those molecules, because how does RNA polymerase know, and it produces a transcript, the RNA molecule. So how does RNA polymerase know where to start and where to stop transcribing? How does it know where to start? Transcription. This is translation. So this is the process of transcribing a gene to produce the RNA molecule. So this is a good review. We've got proteins like TATA binding protein that bind a spot in the promoter of a gene and then pull in RNA polymerase and hold it there where it's supposed to start transcribing. RNA polymerase starts transcribing, and then it knows to stop transcribing. How? So eukaryotes, let's focus on eukaryotes. And we'll pick this up, I'm going to answer it quickly, but we'll pick this up again on Wednesday if there are more questions. In eukaryotes, it hits that AAU-AAA sequence that's present in the DNA, and that's where transcription, it's approximately where transcription stops. The point of this is that regardless of whether or not this gene is wild type or mutant, both of those DNA sequences have the poly A signal sequence, AAU-AAA, in them, and they also both have the promoters, which is where those proteins bind to start transcription. So in those two places, these two chromosomes are identical, which means that transcription happens, the RNA molecule gets produced regardless of what that mutation is, whether it's there or not. You generate a transcript that contains the mutation, or you generate a transcript that's wild type. The only effect of this mutation is on translation. So transcription's identical regardless of mutation. Then we see the effect of the mutation during translation.
Is there a mutations like upstream,
Instead of,
like directly, where the polymerase starts to pick up?
well, so the question was, is there a reason that the promoter is upstream of the transcription start site? And the reason for that is because you don't want to have the proteins that are directing the transcription start site and RNA polymerase to be blocking RNA polymerase. So if the promoter was here and RNA polymerase was here and was trying to transcribe this direction, your proteins would be blocking RNA polymerase from moving in that direction.
And one more thing, is it safe to say that every mRNA starts with methionine?
It's safe to say that every protein starts with methionine. Yeah. Yeah, every protein. Now, proteins, after they're made, sometimes get post-translationally changed. So when you actually look at the sequence of a protein, sometimes it's methionine that was originally there, got removed. That's a whole separate thing. We don't have to worry about that. That's biochemistry or cell biology. Take those classes, please. Okay, so come with questions for next class, and the top point will be probably very important to you. We are still looking at those group shared definitions class notes, so they're going to get graded again on Wednesday. Please make sure that you've attended to those. I've got all the keys for these two assignments online, so take a look at those, bring questions on Wednesday.